good afternoon everyone. Welcome to Liquid Brain. So today I want to take a look at MAFtools and how we can use it to summarize, analyze, and visualize MAF file. Specifically, MAF file stands for Mutation Annotation Format, and this tool will allow you to report your somatic mutation data in your cancer patient. So before we go deep into the RStudio and the package, perhaps let's take a look at what is actually the MAF format, so mutation annotation format. So based on the GDC manual over here, it's a text delimited text file with aggregate mutation information from the VCF files and are generated on the project level. Okay, so the actual detail on how it is being generated and what information to include or not include is actually quite complicated. We're not going to look deep into the file format, but rather we're going to go into the MAFtools and how we can use this data to actually visualize and analyze our data. So you can look on screen right now, it's an RMD file, so I do not write most of the file myself. I actually download it from the author's GitHub page. So what I'm going to do is I'm going to include both the link to the original script file as well as the script file that I modified so that you can choose which one you want to download. But if you download mine, this way, so for mine, I actually included a little bit more details about the subsets and the merging, while the original script has a lot more details on how you actually analyze them. So I removed some of them just to help with the flow of the detail, so they might be missing when you compare to the original. So when you're done, you can open it on RStudio and it should look somewhat like that. So if you get very confused, it's because this is actually a markdown format. So markdown format has two sections. Just remember that everything outside of the shaded area are non-executable, while everything inside the shaded area are executable. So this is the R script. Everything else up there is just text for you to actually understand what it's trying to say. So I find it very useful because this chunk allows me to really focus on what each chunk needs to do and need to accomplish and help me to communicate better, which is why I almost always use them. And you realize that a lot of people in the tutorial script or vignette actually use a markdown file for the communication. And I believe the website that you see right here is actually generated from a markdown file. Okay, so once you're done, let's take a look at the first section, which is how to actually import and format the MAF file, how to subset them, and how to merge them. So just to make sure that the MAF field requirements are ready when you import them, your MAF file in your TSV or in your whatever format CSV that you want has to contain all this information. So we know what gene are they, what chromosome they belongs in, and the location and variant type, such as the insertion, deletion, and so on, is complete. So beside actually downloading an MAF file, you can also generate them directly from VCF, as we talked about just now. You can use this tool to actually do the generation, or you can also use ANOVA over here to actually generate MAF based on the input that you want. But the easiest way, of course, we just try to use whatever people have generated, specifically from TCGA. Okay, so for the reading of an MAF into the project, in this case, we need the MAF file with all the requirements that we talked about just now, the Hugo symbol, location, and so on. We also have some optional information such as the clinical data and the copy number data, which can be included, but it's not strictly necessary for most of the analysis that we are going to do. So what and how we're gonna actually include them is actually use the read MAF function, which I'm gonna show you right now. So first of all, before we do anything, include the MAF into environment using a library command, otherwise it will not work. And the first thing here is to actually redirect the system into where are the files, right? So there'll be two files here. We're loading in an MAF file and an annotation file as the clinical information, which is of course an optional information. And if you actually look at LAML-MAF, is simply a file directory, or it's a file URL, and you actually just redirect the RStudio to this file. And once you're done that, and with these two information ready, you can just run read MAF. This is equals to this, and this equals to this, and you will load in an object as LAML over here, and you'll be able to see in your environment like so. Okay. So once you're done that, I just include another example of how you can actually build the same thing without using the clinical data. In this case, BRCA. So I believe LA ML is actually leukemia data, BRCA is breast cancer data, just different data set, different sample. So you can also subset the LMF object into something called MF data view, where you can take a look at the data or the information contained in the MAH object, or you can actually have a look at the information of the gene summary including the object. But strictly, to be honest with you, I prefer to just go into click to the object and actually just explore them one by one. And you can also know how to actually subset them by just typing the name of your object, LAML, and just type add. And you can actually go through one by one, for example, summary, and you can subset a column, for example, out of the list that's inside MF object. So it's quite straightforward because the data here is quite small, so it's quite easy to navigate around. Okay, so most of the time we don't actually want to do that because we don't always have a MAF file so for example, if you do your own experiment, obviously you will have your own MAF generated from VCR, but most of the time once we generate it, we want to compare our information against other study. So in this case, we're going to use some the ready databases, ready-made database from TCGA. So we're going to use a library called TCGA BioLink, where we can actually get the project summary from all this thing. So most importantly is just that the file type will be equals to the MAF file format, and we can download it directly using the GDC download and pipe it into a data using GDC prepare. And once we're done that, we just say that it's a TCGA data type. I'm going to use a read MAF again to read this data object into a new MAF TCGA, which I'm going to show you right here. Yep. So you can see that most of the information is very similar to what we have just now, almost exactly the same, just with different number of data and different number of genes, obviously, and different number of sample name. But the building of it is exactly the same, and which is important because they have to be the same for rest of the for the rest of the analysis to work. The structure has to be the same. Okay, so you can type LAML directly to look at just a summary of the MF object. What are the deletion, insertion, mutation they have, how many times it appear, and what are the mean and median that you have. Doesn't make a lot of sense here, but you will get more information later with the visualization. Okay, so in this case, these are just a little bit more information on how you can get the sample summary, the gene summary, the clinical data, the fields, and so on. And you can write a summary file into your current directory. But I don't find them particularly much more useful than just clicking the object and try to exploit one by one over here. But if you do need to, for example, write a certain custom function to do a certain thing, it'd be useful to actually know all of this function and what can they actually get you. Okay, so once we have import our MF properly, it's actually time to subset them. So how to subset them is actually very similar to what you do with most of the data object over here. So what we do is just use a subset MAF, and we can choose the gene that we want, we can choose the query that we want, we can also choose the query with a little bit more not just logical, but more of a larger and lower than situation. So a little bit more granularity in the way that you want to filter your data. We can also filter based on the barcode of the patients or patient group, the fields that we want to include, or the chromosome that we want to do. So this will be important. For example, you want to compare chromosome 2 and chromosome 17. You can actually subset 2 and 17 here and compare it against maybe 3 and 15 later. So you can actually just subset a big MF object into 2 small ones, and then you can do the comparison later, which in the end of the video, I'm going to show you to do that. Okay, so you can also get all this summarization while you're doing the subset above, because we're not building it into a data object, but these are the printout, and you can check if that is what you want. So once we're done that, we can again merge them together. For example, you have 2 different studies, or you actually read multiple TCGA different data project, and you want to merge all the data together for downstream analysis. A merge MAF will be able to actually help you over here, and it's just putting in the name of your MAF, and they will just join them together. Just beware that when they join, they might not check for example, duplicated genes, duplicated sample, duplicated symbols, or some other way. They simply just merge them together based on the logic that they have. So you can take a look at the detail, for example, source code over here on how they merge, but I'm not going to go through them because we do find some problem last time when we're using this, and the merging does not work properly because it might be because my ML object is broken and so on. But in LAML and TCGA MAF object, it actually works quite well over here. So you can also see based on the verbals, what are the information that they use. Okay, so that's done the first part of the analysis, which is the input, subset, and the merging of MAF object. And then we're going to go into the second part, which is the visualization of the data. Okay, so the most useful and almost the first thing that everybody runs is the plot MAF summary, which is you have an MAF file, what I want to see, what is the general overview of this project. So you can see that I'm using MAF TCGA, and we can see what are the different variant classification, variant type, SNP classes, variant classifications, top 10 mutated gene, variant per sample in different file format that I want to. And it will be very useful to identify something very obvious. For example, what are the top 10 mutated gene? So again, if just to give you another example, let's try our LML over here, and you can again see that these are the gene that changes a lot. These are the transition or transversion percentage in each, and so on and so forth, and what are the average or median variant per sample and the distribution. So it's really helpful to help you to understand a certain thing, and you can again go get individual plot individually as shown over here. So this is the bar plot where we have a top 15 or 25 genes over there, and what is the type of deletion, mutation they have inside each of them. So you can again just change the MF object to, for example, BRCA, and you can actually have a look at what are the mutation for each gene and which one occur more than the other. This one not particularly well. I'm going to go for the TCGA over here, and you should, yeah, it's a little bit better, but it's still, most of them are missense, while AML has a lot more variation or variety in terms of its mutation type and genes and so on. I think which is why the author put it as example data. It just looks a lot nicer, isn't it? Okay, so besides just putting on the bar plot, we also want to do something like an Oncoplot, which is oncology plotting, stuff like that. So in 193 sample, besides knowing the genes that have been most mutated, we also want to know, like, in this mutated gene, what are some differences between this sample that I have. So something important, if you look at FLT3, when you have FLT3 red color is in-frame insertion, it doesn't cause or co-occur with other kind of mutation that we have. However, for TP53, if I'm not wrong, when you have it, also you don't have other one. So maybe it's DMNT3A. I actually have an analysis section down below. So if you have this mutation right here, you can see that it also affects some other mutation in the same sample. So there's some co-occurrence of that, and only Oncoplot here will be able to show you on top of the, what is that called, the bar chart over there up there. So there's more information here that you can actually try to identify a point. Okay, so there are a little bit more wording here that you can actually read and try to understand, but I'm just going to talk through it because I'm saying the same thing as the words, and research show it's not good to show the word and say the same thing because people get distracted and so on. So if you do want, pause the video and you can go back to the script and read it yourself. So the next one I'm going to do is the transition and transversion plot. So what that means is transition when you have A changes to a G or mutated to a G or G to the A, while C to a T, these are all called transition because they are changed to the same type of nucleotide, purines to purines and pyrimidines to pyrimidines. However, if you have a purine jump to a pyrimidine or pyrimidine jumps to a purine, we call them transversion. Okay, so transversion I believe is a little bit more serious than the other one, or not, depends on the translation, the location that you insert in, but these are just a different kind of plot. And you can actually see that in this sample, which is the LAML sample, most of the mutation here is C to T, which is a kind of transition, and you can see that the transition occurs a lot higher compared to the transversion as well. So it might be because of a certain gene, certain location, but transition is definitely more significant in most of the sample as compared to transversion, which is what this plot is supposed to tell us. So once we have figured out an overview, now we are ready to go into individual genes analysis and details. So which is what we're doing, which is here as a Lollipop plot. I love the name, to be honest. So a Lollipop plot actually shows the mutation rate of the gene and where they are and what kind of domain to the occurs on. So for example, TP53, you can see that these are the location of the mutation, and these are the type of mutation that they have. And if you want to see that, for example, what is the other one that is the most mutated one over here, you can just change the name over there. Oops, change the name here, and you can actually run it again, and you can see that most of the genes are mutated right in front of this domain and right in the middle of this domain. And if you do have a very high mutation rate, for example, you can actually label them so that it actually shows you you are a better person in terms of the data visualization or presentation of data. If you want certain thing to be labeled, you can just check on the location of those mutation and try to just put label position on, and you will have the mutation rate done. While some of the other percentage mutation, for example, is already being put here, and you also have the RefSeq sequence up there as well. So these are the domain that have been mutation hotspot within a certain gene. So of course, if you don't want a mutation, you can also use the plot proteins just to see the domain, and you can put it as a RefSeq ID so that you can make sure that it's showing you the correct protein and so on. Okay, so the next one is, again, we're moving back into the overview. So it's called a rainfall plot. So what does this work is that you have two axes. Once you've done that, the x-axis is the location of the chromosome 1 to chromosome 26, and in the end is the x and the y, because that's 21 to 23, and in the end is the sex chromosome right here. So in the y-axis is a log distance between two mutations, right? So remember, it's a log, so you can see most of the gene occurs around here, which means that every single mutation has an average length of around 10,000, if I'm not wrong, log 10, yeah, about 10,000. So every mutation has 10,000 distance in between them, right? So one mutation, 10,000 nucleotide later, you have another mutation. However, in breast cancer, there's a very special case where between the chromosome 7 and 8, you can see there's a location where there's so many mutation, and again in 11 to 12 and 15 to 16. So this kind of mutation hotspot could be a causation of the cancer or relation of the cancer, depends on your situation. But breast cancer has been significantly this situation or the phenomena or the patterns has already been detected in breast cancer again and again and again. And if you do have mutation in these three sections of the gene, I'll suppose you get a check on whether your breast is having cancer or not. So this does not happen, for example, in some other cancer like LAML. So LAML actually, in comparison, has a lot less mutation compared to BRCA, but they just have mutation in the worst possible place, which actually result in them getting leukemia. I believe it's not as serious, so maybe they live much shorter than breast cancer, which is why they die early, and there's no time to there's no time to accumulate those mutations compared to breast cancer, because if you're leukemia, I think you die much faster. I don't know, don't quote me on that, I'm not a clinical practitioner or something like that. So once we are done, the next one is actually the mutational load. So here it's actually called TMB, so tumor mutation burden, if I'm not wrong, or some sort of things like that. It just compare per megabyte, which is per mega base pair or something like that, per million base pair, how many average mutation are there. So 1 actually means that you have 1 mutation per million, and 10 is maybe 10 mutation a billion, and so on. So you can compare between different cohort. For example, you have BRCA, which is the sample that we put it in. You also have some other different kind of cancer. For example, LAML has a least number of TMB as compared to the others. Actually, we had also looked at the rainfall plot right here, while some of the SKCM has a lot higher. And again, if you just want to change out this kind of thing, you can also do that and see if it works for the TMB over here. Yep, okay, see? So you have something right in the middle. Oop, I should not have used the same example. So I should just do that. So yeah, you can see in comparison, the example, which is the TCGA right here, is slightly below the BRCA, but also ranked right in the middle with all the other cancer and all the other cohorts and so on. Okay, so the next one we're going to look at is called the variant allele frequency. So if you remember like your first grade genetic class where we talk about allele is the same gene on the two different on the pairs of chromosome from your parents, right? So if you have equal pair, if you have, for example, a pure sample, you have 50% of gene from your mom and 50% from your dad, they should be identical, which is why the mutation should be exactly the same with each other. But in cancer cells, sometimes it goes really wrong. So you can see that. So ideally, everything here should be 50%, right? But you can see in most of the example, they're not 50%, where most of the time one has a lot more than the other. Depends on the definition of genotype over here. And you can actually have a see at their equation on how they calculate the percentage right here. But usually what I will look at is the more deviated from 50%, the worse. For example, NPM1, very far away, while TP53 is not too bad, and TE2 here is also not too bad, something like that. Okay, so the next one we're going to look at is the copy number data. Copy number is actually generated by some other external program called, in this case, GenStick, Genomic Identification of Significant Target in Cancer. We're not going to look too much, but we know that somatic copy number alteration of gene could actually drive cancer growth, and is something that we find correlation with. And you have a group that you're looking at, it's important that you might want to look at the SCNAs and actually try to see if there's any pathway or mechanism that's contributing to this variation, and how could they actually lessen them to solve the cancer, cure the cancer, or just simply slow down the cancer. So those are very important. So once you get those data out, again, they'll be in TXT file. You can load all the TXT file as a system file, and then use a read GenStick function to read all the file that you have specified above into a GenStick object over here. Yeah, large GenStick object, which is actually quite similar in terms of overall structure compared to our MAF, but they contain different lists inside the object over here. Okay, so they're not the same type of object, and when you run the above functions on this object, it would actually not work. Okay, so once we're done, we can actually look at now is the copy number variation, whether it is a kind of amplification or deletion, which means they have more of a certain thing at this certain area, or they have less of a certain thing in this certain area. And you can see that in chromosome 1, chromosome 11, and chromosome 21, these three site has a lot of amplification of this thing, and this 1, 2, 3, 4, 5, 6, 7, 8 has a lot of deletion of all this thing based on the copy number that is generated from the GenStick. Okay, so beside doing the positional plot, you can also plot it right here, so that it's the number of sample and the percentage of gene. So usually when we look at graph, everything on the top right corner is worse and they're more significant. Everything on the bottom left corner, they're not as significant. So, and you can see that the bigger means there are more Q value. If I'm not wrong, that's a significance calculation. So that you want to look at 1, 2, 3, 4, 5 over here, or the rest you do want to look at them, but they're not as significant compared to the rest. So that's kind of conclude the copy number. So next one we're going to look into, the next one we're going to look into the Oncoplot of the copied number. Okay, so we're going to use the same GenStick format, and rather than just looking at the significance and so on, we're gonna have to look into a little bit more details on the sample, and each sample, how does it work in each of the site with amplification or deletion. So this is what we end up with. And again, something interesting is this person has 1, 2, 3, 4, 5, 6 different deletion, and this person has 1, 2, 3, 1 deletion and 2 amplification. So we do not know the mechanism yet in this case, but this will be something that you want to take note when you're doing the, what is that called, the analysis of your overall data and a comparison. So this is something that I'm not super qualified to talk to, but is CBS, which is circular binary segmentation, which actually partition the genome into segments of constant total copy number based on DNA microarray data. I believe is a kind of a normalization for the calculation where we have above, which is just a better way of doing it, but I'm not super sure about that. But you can also see that they're very similar to the way that we present data above, which is these are the points that we want to look into, and the higher away they are from the mean, the more interesting they are in this cohort, and we want to take a look at them specifically and so on. So I'm not super sure. So if you are an expert, maybe leave a comment down below on what they are and how do they do. And again, if I do anything wrong, please go ahead and leave a comment down below. I don't want to just mislead people for the longest amount of time. And after that, I'm gonna go into somatic interactions. So what is a somatic interaction is there's certain things and a certain a mutation that occur together, and there's certain mutation that never occurs at the same time. So somatic mutation variation, or SMV, is supposed to show us that. So for example, let's look at the purple color here, which is co-occurrence. So ASXL here always co-occur with IDH2, with RUNX1 and TTN, while TB53 and FLT3 has never occurred on the same person, which means that if you have a mutation in FLT3, you will not have it in TB53 and vice versa. So here as well, IDH2 and IDH2, I don't know if they are the same thing. They could be the same thing. No, I'm looking at the wrong column, sorry. And so the IDH2 and the NMP1 also does a mutually exclusive, which means that if this happens, that will not happen, and so on and so forth. I think the most interesting one here is this one, DMM3A, which is the highest mutated gene in all the sample, and you can see that always co-occur with SMC3, the IDH1 and NPM1. So there might be some interaction that we want to look into going forward from that. But these are the way that we can understand what kind of thing happens together and what kind of thing never happens together, and that is called the somatic interaction graph or interaction plots. Okay, so once we're done with the somatic variation, we can also detect what is called a cancer driver gene. So if you think of a driver, it's something that drives you to some place. So a cancer driver is a particular gene that causes cancer. So how do they cause cancer? There's an algorithm called Oncodrive class. We're actually based on hotspot positioning and enrichment, and a lot of mathematics to make it work. But what we do is we just accept that they're correct and let some expert to check with them, and what we do is that we can just take a look at the analysis, the result that we have, and you can see that NP1 here very highly significant, and we have a lot more other information over here, and we can try to look at the mutated sample, and we can look at these other gene that we might want to look forward into as a potential cancer driver. So we can do that either through downstream mutations, simulations, and perturbation, or some other way that we want to look into. Or simply just check if this cancer driver actually is the same as what people has found previously. That will be the easiest way to do it. Okay, so beside using a table, which is like very ugly, so we can also do a Oncodrive plot. So we can actually see that again, everything on the top right corner is usually more important. Everything on the bottom left corner, they're usually not as important. So IDH2, you can see, is the highest mutated. IDH2 here, 1 and 2 is the highest mutated. Where my IDH1? Yeah, it should be overlapping with 1 and 2 over there on the top right corner. So yeah, I would make this a little bit bigger and then make the scale a bit smaller, but you know, that's the default. You can go ahead and run that. So the next one is the summarizing of the PF domain. Again, I'm not super qualified to run that, but usually when I look at a plot like this, everything on the top right corner is more important, and these are the protein domain, I believe, that has been enriched. Yeah, so these are the protein domain that you want to look into, whether they have a specific correlation or causation situation with the sample type they're running on, and this is something you can look into with the pfam domain summarization and plots and so on. Okay, so the next one is survivor analysis. In simple state, with this, so two different groups, which one survive longer? That's how I would like to summarize survivor analysis. So for example, FLT3, you have it or not, does that affect your days to overall survival status? Do you live longer with or without the gene? Mutants mean they have a mutated gene. WT means wild type, means they don't have the mutated gene, and this is the number of patients in each. And you can see that in this case, the p-value is around 0.113, which means that we can't tell whether the difference between the two groups is by chance or not. So we can't say that FLT3 definitely affects your your survival or not. It's hard to know. But for example, TP53, if you can run it here, you can actually see that the p-value is 0, very, very low. And we know that if you have a TP53, almost confirm that is gonna, if you have a mutated TP53, most likely you are gonna die. So this is what this graph is trying to show you. Okay, so the next one is, of course, when we do survival and we do analysis, we do not want to know a single gene and a single cause and mechanism. Gene expression and interaction is very, very complicated, and we want to know if two genes works together. If they mutate together, is there higher chances? Are there specific situations where these two genes mutate together is much more dangerous than one mutated by itself? So the survival group here is actually trying to solve that. So you can actually look at the gene combination. For example, if you have a mutated FLT3 and DMMT3A, most likely you will have a much lower survival rate, if I'm not wrong. So these are all the gene combination that you will want to be look out for. And if you have this mutated DNMT3A gene, that will be very dangerous for you. So maybe you can go ahead and scan for the mutation in your body directly. And these are the way to visualize what we have found just now, which is these two genes together, very dangerous. And we can say that, yeah, they are very dangerous. Not as bad as TP53 though, but still, the gene set here, it's surviving a lot lesser than the wild type. While if you have the second one, which is DMN33A and MP1, it's still bad, it's 0.07. So there's a high, it's about 7% chance that this is by coincidence. And if you do have these two mutated genes, I would suggest that you do some more testing as well on your body, or just check for other confirming variable, whether you have cancer or not, or some other pathway mechanism that we can look into, particularly with literature review. So once we're done that, the last part of the script is actually about the comparing of the MAF file, or you just have two different cohort, different experiment, and you want to compare them to, or even you have your own and you have something online and you want to compare them, this will be very useful for you. So how we do that is, again, we're going to create two new MAF object first, without the clinical data, without the copy number data, and this will be just relapse and primary. So if you're not from cancer science, relapse means you have cancer, you cured the cancer and it comes back, right? Primary means you have the cancer on the first time. So relapse usually what we do not want, because when we treat people with cancer, we do not want them to have cancer back. So relapse is really, really bad, which also explains why, if you look at the summary, the primary has 124 people, while relapse has 58, because some of them cured completely, some of them maybe did not make it through the primary, so they die halfway, sadly. So usually relapse has much less number of samples that we have. Okay, so the easiest way to compare is forest plot, where actually you plot them against one another with the odd ratio. You can have a look at my previous video on how to understand forest plot and how to actually customize them. But you can see that these are the few genes that you want to look into, and they are significantly different from two different population or two different MAF files and so on. So something you might want to look into based on the odd ratio and their p-value. Okay, so once we've done that, we can also visualize it beside the forest plot. We can visualize into Oncoplot, Oncoplot in this case, So which actually give you a much better idea on how do they mutated and how do they, what are the mutation in each of the patients that we have. So yeah, something very interesting is FLT3, or almost exclusively on primary. And when you have FLT3 mutation right here, you can see they do not have the rest of the mutation. However, for something like PML, RUNX1, ARDR1, almost three of them almost occurred together on the first three sample. The other one is RARA here. Actually, you can see that they're almost exclusively inside relapse and nothing on the other side. So again, some pathway that you might want to look into on whether they are causing or they are an effect on the outcome or result of having a relapse cancer right here. Okay, so again, same thing, but you do it as a COBA plots, where you can actually see the FLT3 mutated a lot in primary, nothing in the relapse. And RARA here mutated a lot in the relapse, but nothing in the primary. So similar result expressed slightly differently. Okay, so once we've done that, we can also use a lollipop plot to actually narrow down a single gene that we have, in this case WT1, and we can actually see that everything on top is the primary, everything below is a relapse. So WT1 doesn't actually show that much difference between the two. Maybe we should try something like RARA, for example. So you can see RARA only appear on a single site, and maybe we can do FLT3, FLT3, so it will only show up on the top side as well. But you see that everything in the primary, some of them also occur in the relapse. So we don't know what happens, but this is the result that we get. Okay, so that everything just now is just about the mutation data, plotting overview, looking at individual genes, and so on. So we also talk, since we also talk about the clinical enrichment analysis in the beginning, we talk about adding the clinical data into the MAF object in the beginning. There are also some ways that we can actually use them to run our enrichment to see if that actually affects our data and what are the analyses that we can do. So this is actually called clinical enrichment analysis within a certain kind of clinical, what is that called, threads. Do they have a significance between one group or another? So for example, the example here uses FAV classification. So if you don't know what is FAV, is actually, what is that called? I actually don't remember. Yeah, French, American, and British classification, which is, in this case, the higher the number, the better. Sorry, the higher the number, the worse it is. I think I put it here, yeah, right, yeah. So M0 means is you don't have as bad of a cancer, but if you have M5 means your cancer is really, really bad. It's some sort of clinical traits like that where the clinician assign it onto a patient during diagnosis. Okay, so we can actually compare between the M1 to M5, M1 to M7. What are the genes that are significantly different between the two groups, right? So in this case, it's M1 against the rest, M7 against the rest, me against the world, and so on and so forth. And what are the genes that has been significantly enriched in certain situation, and what are the one that doesn't? So again, we can see that we're using LAML over here. So just beware that we are not running on primary and relapse. And the IDH1 comes out again with TP53, and this one always come out DNMT, where it's always in group 5 against the rest. So yeah, something we want to look at is actually M5, because they're not the largest group, and they have this gene happening a lot in this case. So something I might want to look into afterwards. Okay, so we can also plot the enrichment result, which actually show the thing just now, where we have like the enrichment in certain thing and not the other, and these are the gene that we have in M1 to M7 against the rest, and so on. Okay, so once we've done that with all the clinical data, we're also going to do a little bit of drug-gene interaction before we end up the session. So drug interaction is based on this website called Drug-Gene Interaction Database, DGID, I believe so. Yeah, so you can actually see between the LAML and BRCA based on the gene expression and mutations and all the database that the website has done it. These are the possible categories that we want to look into, like clinical actionable, these are the gene that is in this category. We have druggable genome, we have kinase, tumor suppressor, and so on. We also have this one, all the other gene that I do not understand because it's breast cancer, and we have FSDL1 and so on. So these are all the drug categories that we might want to look into and might be useful for the analysis that you might be doing, and so on and so forth. So we can also go for signaling pathway, which is like, is RTK-RAS significantly affected? How about HIPO? How about WNT, and so on, and the number of sample affected for each of the pathway. So TP53 always there all the time, and we have Notch and HIPO and RTK-RAS, which is affecting half of the sample. So yeah, something definitely look into right after this video. So the last one is, of course, just plot it out, and exactly the same just now, just plot it out as this specific pathway, what are the gene being involved and which side of thing they are involved. Very conveniently, the plot oncogenetic pathway also do like super tumor suppression gene in red color. So you can see that these are the CBL and NF1 is actually tumor suppression gene, and oncogene are in blue, and I believe black is there in neither of the category. and so on. So that actually concludes the script for today on a quick look at MAF file. I hope that you learned something about MAF. I believe it's a good experience for me to go through and look at beautiful pictures that is being generated on the data, and actually found something that I have not found beforehand myself. Okay, I hope you learned out something. Thank you for watching, and I will see you in the next one. Bye.